Okay, it's 4 o'clock, so I'm going to go ahead and make my announcement so that we can get services and try to get everybody out in a timely manner. Next week, we're going to start the first of 2 virtual poster sessions. So it's the first time we've tried to do this, but the students who are actually enrolled in FNR 8080 graduate seminar are going, we usually do a poster session down in the Lahotsky lobby at the end of the semester to share their research and what they're doing. We're going to do it virtually starting next week. Those students know that there were 2 of you who still haven't signed up, and you are now doing it next week on 416, Chip Bird and Hunter Morton. Everyone else, and Hunter is here, so he just heard that announcement. I might have to email Chip because I don't see him on here. So just so you know, you guys are doing posters on 416 next Thursday. And I will turn it over to Crystal to start the graduate student sampler we have today. If you aren't speaking, if you will mute your mic, we'd appreciate it. And then you can type any questions you might have in the chat session or unmute your mic after each presenter. Thanks.
All right, so welcome to the NRGSA Seminar Series. For those who don't know me, my name is Crystal Strickland. I'm currently a master's student under Dr. Don Hagen. I'm going to be introducing your speakers today. Today it's a graduate student research sampler, and we have 4 of our own students presenting their research to us. After each one of them speaks, you will have about 5 minutes to ask questions to them. And again, like Dr. Thea Hagen said, you can either ask them in chat or unmute your mic and ask out loud. Our first presenter is Matthew Vaughan. Matt is a master's student in forest resources under Dr. Don Hagen. His research interest is biogeography, vegetation dynamics, and applied fire ecology. He is studying the effects of seasonality of prescribed fire in the Southern Appalachians, which will be the theme of his presentation here today. Matt, you have the floor.
Great, thank you, Crystal. Here I'm going to share my. All right, can y'all see my screen?
Yes, I can see you.
Great.
Yes. Kaminsky says yes.
Great. Thank you, Crystal, for the introduction. So today I'm going to talk about a component of my research here at Clemson, and looking at fuels and fire behavior as a component of seasonality of prescribed fire in the Southern Appalachians. So just to start off and thinking about the prevalence of fire historically, was fire prevalent in the Southern Appalachians? Well, from the best that we can gather using dendrochronology as well as soil and sediment samples, and reconstructing past fire occurrence in the Appalachians, while fire was variable from year to year, on average it was more frequent than it was until the 20th century with active fire suppression. So in thinking about historical fire regimes, we now see that there's the effects of fire suppression, right? So the regenerating desirable species is difficult. Many forests have more mountain laurel and rhododendron, for example, especially in the Appalachians. And so managers often now use prescribed fire in order to elicit desirable fire effects as a tool for restoration. But in thinking of this, what fire regime are we restoring? And does prescribed fire...
Matt, may I interrupt for just a second? I'm hearing a lot of choppiness. Sometimes it's very clear, but I wonder if it has to do with the closeness of your mouth to the microphone.
Okay, I can try moving back. Is this better?
Okay, try that distance.
Okay.
That sounds good.
All right, thank you, Dr. Kaminsky. So I'm thinking of does prescribed fire today, how does it match that of historic fire? So the trend today, largely, this is the graph showing the acreage burns in the Great Smoky Mountains National Park over the last several decades. And as you can see, during the growing season, as that green line in that graph shows when the TPI values are highest is when there's the least amount of fire. Today, we don't have much fire in the growing season. So most of the modern prescribed fire occurs in the dormant season, but managers consider burning in the growing season. So this was identified as a research need in 2018. And so for managers who may want to expand their program
Hi, Matt.
season burning. Yeah.
It's still doing it. Maybe try turning your video off. There may be a connectivity issue. Let's try that and see if just turning your webcam off will kind of clear it up and allow it to come through better.
Okay, I'll try that. How's that?
We can still hear you. It just kind of comes in and out. So go ahead and keep going and we'll let you know if there's still a problem.
Okay. All right. So for growing season burning before canopy closure may offer an alternative to dormant season burning, allowing for increased opportunities to burn. But it remains to be seen whether growing season burns can accomplish the same objectives as effectively as traditional dormant season burns, particularly for managers who aren't familiar with burning in the growing season. Trying to advance the slide here. I apologize. It's not letting me. There. How and according to which factors does prescribed fire vary by season of burns? For this project, we're defining growing season burns as those in the early spring green-up period before canopy closure. And so I'm going to focus today on the first chapter of my thesis. And some of the questions that we're going to be looking at today are, one, how do fuel moisture and meteorological conditions on days of burn differ between dormant and growing season burns? How does the proportion of area burned within plots differ? And then finally, how does surface fuel consumption, fire intensity, and the relationship between them vary between seasons of burn? So just as an overview of where my study is, we are in the Andrew Pickens Ranger District and the Chattooga River Ranger District of Sumter and Chattahoochee National Forests, and we established pre-burn plots. We collect day of burn data, and then we also collect data after the burn. So this just shows a progression. I'm going to try to go through this faster of the different burns that we've had. So a total of 4 dormant season and 3 growing season. And this figure here shows that the range of dates for growing season burns was less than for our dormant season burns, all in about this time of year in April. I got some burns. This just shows the schedule of collecting that data. I can go into more detailed methods later if you have any questions, but just as a quick overview, we used Brown's fuel load protocol. We measured litter and depth consumption with nails, and then we also collected fuel moisture on the day of burn. And then finally, we deployed thermocouples to measure fire behavior during the burn. Some results. Going back to our first question, looking at fuel moisture and meteorological conditions. This table here summarizes the differences by season for each of these variables. So the values bold here on the right-hand column are those that had a statistically significant difference. So we used a parametric test here, and we compared all these different variables. The results here indicate that growing seasons had a lower fuel moisture than dormant season burns for the different fuel types measured. And then, as we expected, solar radiation and air and fuel temperature were also greater with growing season burns. So we had drier fuels and more sun and warmer temperatures in growing season burns, and we would expect that whereas the growing season burns are later in the year. So then looking at how the proportion of very burns within plots differ by season. This graph here shows that it's a box plot. So it's there's a lot more variability in how much plot areas burn in our dormant season burns. Almost all of the growing season burn completely, and only these outliers here did not. And then comparing the moisture versus that, we just looked at a plot area burn. On our x-axis here we have fuel moisture. So as you go from left to right, it's getting drier. So lower or higher to lower fuel moisture is. And you can see the relationship between the positive relationship between fuel moisture and plot area burn and then by seasons. This illustrates how much more area was burned in our season burns. So then now looking at our response variables of consumption and processes of fire intensity. This table here summarizes with rounds. So we've got both the variability of the response itself. None of these variables were statistically significant in terms of the itself. So like, for example, when our cooler and so forth did not statistically significant for by season. There wasn't very significant differences with the variation of the variability. So the gist of this table is that surface fields did not vary by season, and the variability of that response did not vary consistently.
Matt,
Direction. Yes.
I hate to do this, but we can't really understand you at all anymore. The only other thing I can think, what kind of mic are you using?
Just the mic on my computer.
Okay. And it's very clear right now.
I'm at the same distance away that I have been.
Okay. So now that you're talking to me, it's very clear. I'll leave mine on and see if this helps.
Okay.
Go.
All right. So now for litter and duff consumption, looking at litter as measured via nails, we had a similar result with the variability of litter consumption. And then we did have more duff consumption with growing season burns than dormant season. We had none measured in our dormant season and just a little bit in growing season. So now looking at fire intensity, this is not true fire intensity, right? So thermocouples, it's an index of heating relative to fire intensity. And this graph here just shows the mean and the standard error around the mean by treatment. And then on your y-axis is the heating of the thermocouples. So, or the degrees Celsius times seconds. So clear both greater mean as well as the variability around that mean for growing season burns. And then kind of digging in deeper, this graph here shows over the course of the day of the burn. So on your x-axis, elapsed time since start of burn. Y-axis is for all of the temperatures recorded above 60 degrees Celsius. It's a 1 hour rolling mean for each treatment. So the thick lines are those for the entire season, and then lines in the background represent individual burns. But this graph shows how in the afternoon, as you're reaching your lowest fuel moisture, you have also great variability around the mean for your growing season burns. Whereas with dormant season, it's lower and more consistent variation around that mean. So just some takeaways here. Growing season burns had a greater degree and variability of heating, but not necessarily greater fuel consumption than dormant season burns. And so this may have implications for managers who are looking to restore fire-suppressed landscapes. And in terms of fuels-related objectives, there's no indication that burning in the growing season, or like a shoulder season, if you will, can't accomplish these objectives as effectively as dormant season burnings can. So this work is ongoing, but I just wanted to present an update on this. So with that, I'd like to acknowledge the funding for this project, and my committee and all of the grad students, undergraduates, folks in the Forest Service and Nature Conservancy who have helped in this process. So thank you. With that, I'll take any questions.
Hey, Matt, this is Dr. Kaminsky.
Hey.
I enjoyed your illustrations. As Crystal said, we didn't hear much of it, but I did note that you were using non-parametric methods, a Kruskal-Wallis test. And based on my statistical training decades ago, reporting a mean and a standard error after a non-parametric test is unjustified because you're using a distribution-free statistics. I would suggest that given that it's just a one-way analysis, of variance equivalent to a Kruskal-Wallis, to run it both ways with ANOVA, the parametric ANOVA versus the Kruskal-Wallis, you get the same outcome. Use the results from the one-way analysis of variance, because that would justify you presenting a mean associated with a standard error.
Yes, thank you. I appreciate that. Yeah, the mean values represent, the mean of the response, but then the statistical significance was not based on the mean. It was based on a rank-based approach. So that's a good point. I'll take a look at that.
Yeah, you know, when you use the non-parametric, you're supposed to be reporting ranked values, but they're not meaningful to people. So, you know, I have always advise my students, you know, run it both ways with the parametric and the non-parametric. And if the parametric yields the same as the non-parametric, report the parametric results.
All right, thank you.
Talk that over with Dr. Bridges.
Okay, will do.
Do we have any other questions for Matt? Okay, well, thank you so much for presenting. We'll move on to our second presenter. It's going to be Alex Jensen. Alex is a second-year PhD student advised by Dr. Jakowski. Alex is from California, but he really enjoys life in the South. Today, he'll be giving an introduction to his research on coyote ecology in South Carolina. Take it away.
Thank you, Crystal. Okay, I'm gonna leave my video on, but y'all just yell at me if there's some issues or anything. Okay, we good to go, Crystal?
Yes, go.
Okay, great, thank you. So a little bit of context. I'm a second-year PhD student, as Crystal said, working with David Jakowski. I haven't done one of these seminars at the department, so I'm framing this as an introduction to my research and just a general update on what I've done the last couple of years. I'll start with some background, kind of focusing on one of my chapters, looking at coyote movement using GPS collars, and then at the end, I'll touch on some other projects I have going on as part of my dissertation. I just want to start with this video. It went viral a couple of months ago. You might have seen it. It's showing a coyote and a badger, and this is underneath the highway in California. And so let's just ignore, right, how cute this is for a second, and like how cool it is ecologically that two predators are moving together, and think about, you know, how these passages under highway might facilitate movement for carnivores like this. So that segues into this rapid and relatively extensive range expansion that coyotes have experienced in the last 100 years. So historically, they were constrained to the middle of the United States and the western part of the United States. But in the last 100 years, they moved east. And really, it's in the last 50 years, they've colonized the Southeast. And the first record in South Carolina was in 1978. And so it's thought that one of the reasons this expansion was so rapid, particularly in the southeast, was that just a lack of other large carnivores like red wolves and pumas, which we extirpated, in the last few hundred years. And so this sort of a large predator vacuum. And so anytime a large carnivore, a large predator, relatively large predator like coyote appears in a new area, we want to know what their ecological effects are. So that's some of the background. And so in the last 10 years, there's been a lot of research on coyotes, and particularly looking at how they're interacting with deer in the southeast. It's been shown they're effective fawn predators, although the amount of predation attributed to coyotes, or the amount of mortality attributed to coyotes for fawns, varies across the southeast. And South Carolina has some of the highest rates of predation due to coyotes on fawns. And so understandably, there's been a response from hunters as well as government agencies to want to control coyotes to help to reduce their impact on deer populations. But it seems like studies have shown that even with intensive removal, intensive trapping, let alone opportunistic shooting by hunters, that coyotes are here to stay. And so we want to know, how we can best manage coyotes, deer, and the other wildlife, for years to come. And so some gaps in knowledge. What I'm really interested in, for coyotes across their range, particularly in the southeast in relation to deer, is how selective they are as hunters. Are they actually really keying in on fawns as a resource during the few months that they're available? Or are they just going about their daily movements and happen to find a fawn, get it down, and they eat it? That's one of the questions. The other one I'm really interested in is how does alternative food might influence that response to fawns? So that could be important for managers. If you're trying to protect fawns, you give coyotes a lot of rabbits, their stomach full of rabbits, and they're probably going to eat less fawns. And so the core research question I'll talk about today is whether coyotes are keying in on fawns, and if so, which coyotes do. My two objectives then are quantify how coyote movement changes in relation to fawn availability and vulnerability. And second, better understand how alternative food availability influences that response to fawns. So the first thing you should know is, I kind of touched on this already, but fawns are only temporarily available, it seems like, to coyotes. So on that picture on the left there, that's our vulnerable fawn. During the first few weeks of life, the coyote's anti-predator strategy is to camouflage, bed down in some vegetation, not move and not make a sound. And so hopefully it's just predators don't find it, don't smell it, and it's left alone. When they get a few weeks old, that one on the right is probably a couple months old, they're bigger, they're faster, they're starting to move with their mother. And so predation rates on fawns go down significantly once they reach even a few weeks of age. And so that blue line here in this graph in the center is showing this fawn availability. So fawns are starting to be born around now, actually in April, and they'll be born all the way, sometime in late July or mid-July, probably the last one that tends to be born. And so there's this peak in May when lots of fawns are born and lots of them are vulnerable to predation because they're just a few weeks old. And so what I'm really interested in is what response are we seeing from coyotes? Are we seeing this response that tracks fawn availability? Are we seeing coyotes seek out fawns? That would be that top sort of hump-shaped curve. Or are we seeing that bottom response where they're just going about their normal movement and there's no relation in coyote movement and fawn space use? So how are we doing this? Put it simply, we catch coyotes, put GPS collars on them, and track their movement, right? And so I have two of three capture seasons under my belt now. We've captured 49 coyotes so far, 22 last winter and 27 in the last few months in 2020. And here's one, this successful capture, and there it goes running away. Hopefully that video is going for you, not too choppy. That's a great release. It's got the collar on, it's gonna start sending us data, and it's happy to get away from us. Good. So where's this all happening? About an hour and a half south of here, south of Clemson in McCormick, South Carolina, McCormick County. It's primarily managed timber, working on 15,000 acres of private land. And what's cool about it is it's really heterogeneous. So it's managed timber, so we got some clear cut areas, some young pines, and even mature thin pine stands. So it's kind of patchy. You can see that here in this satellite imagery. You see that those clear cuts, those more bare areas, even some pastures mixed in. What I'm about to show you now is coyote movement from last May of 2019. So from the first year's cohort from last year. Purple dots are the GPS points. And those purple lines between the points are just connecting the dots. So not necessarily where the coyote was, right? They don't travel in straight lines like that, but it's just connecting those dots so you can follow the track. So I'll start that going. So you'll notice two things. First thing I think is, right, it's kind of it's staying in a similar area. So this is all of May of 2019. So staying in this area, sort of restricted by some highways, maybe some roads. Second thing you'll notice is keep coming back to this central location. Probably most likely is a den there, probably raising pups, helping to raise pups. So what I'm really interested in with this movement is not just that it has a den, but where is it going outside of the den when it's making these exploratory movements away from this den area? What kind of habitat it's selecting for? And then thinking back to when fawns are available, right? May is peak fawning. Are they selecting habitats where fawns are most available to them? And so what we get from that data is this sort of heat map that you might see on the left. So you can think of it as home range or intensity of use or a heat map, right? Where those orange areas are areas where we're getting lots of GPS points. Coyotes are using those areas a lot. And so I'm interested in how those areas are overlapping with fawn habitat. And so what I'm going to be doing is doing a weekly analysis of those home ranges for coyotes, of those heat maps. And so seeing, what is their weekly space use throughout fawning season over 10 to 12 weeks or so. And then asking the question, how does this movement change in relation to food availability or fawn specifically? So what we need though is some data for the food items, right? And so great, we have some Fawn GPS data, which is awesome. Thanks to Mike Muthersbaugh, another PhD student in David's lab. He's working on deer in the same study area, which is really one of the special things about our project. We have coyotes, adult deer, and then their fawns as well, all with GPS collars in the same area. And so that's really great. So thank you, Mike. And so now we end up with this fawn movement data, another heat map. Instead of just one individual fawn, though, what I'll do is combine probably about 100 fawns Mike will end up with at the end of the 3 years, and I'll make a study area wide heat map of fawn space use. And so the question then is, how does this coyote data overlap with fawn data? The coyote data is changing week to week based on individual coyotes. I'll have a sort of a static fawn layer, and I'm looking at the overlap between those two layers. Okay, so what do I mean though when I say overlap? So when you think about heat maps, you can think of it also as three-dimensional, like mountains and valleys, right? So the coyotes on the left, fawn on the right, those peak areas, those orange and red areas, those are areas where we're getting lots of GPS points where fawns are using them a lot, coyotes are using them a lot. Those valleys, those lower areas, green in these examples, the animals aren't using those spaces very much. So what we then do is essentially we look at the volume of intersections. So we overlay those two three-dimensional charts or graphs. We just measure the volume of intersection at each point, or at least 30 by 30 meter cell across the landscape, and look at how it intersects throughout time. So again, so the core part of my research here, or for this chapter, is how does coyote space use overlap with fawn space use, and how does that change throughout fawning season? Right, so we expect the highest overlap in this middle area when fawns are most available and there's the most fawns vulnerable on the landscape. We expect if coyotes were keen on them, that's what we'd expect to see. So again, so that's the first objective. Second objective, again, as a reminder, how does alternative food influence this response to fawns? So now we're adding another layer in. We've got the coyote data, we've got the fawn data, and now we'll add in some other layers of alternative food items. And so we aren't putting GPS collars on everything, unfortunately. So we're using wildlife cameras to track the availability or monitor availability of other food items like rabbits and small mammals. And so again, we end up with these similar spatial layers of heat map across the study area. We have the coyotes, we have the fawns, we have the rabbits. Now we overlay them all. And one prediction we might make here is that in areas with high rabbit availability, you might see coyotes kind of keying in on rabbits instead of keying in on fawns. That's I don't know what we'll see necessarily. I think it's a little bit complicated, but that's one prediction we'll make. Okay, so that's sort of the focusing on one core part of my dissertation. Here's some other chapters. I'm really interested in coyote diet, sort of relating to fawns in particular, looking at what they're eating throughout the year. and how does it relate to availability of fawns, fruit, rabbits, small mammals, etc. Another chapter, so another gap in knowledge in the Southeast in particular is how coyotes are influencing other native carnivores like bobcats and foxes. So I'll use the cameras that we have out. We have 90 cameras in an array across the landscape. Look at space use and time of day activity of native carnivores and how it relates to coyote space use and activity. And lastly, I have a general resource selection chapter. So really just interested in how coyotes are selecting space, what drives their space use throughout the year. So during hunting season, are they avoiding people? Are they maybe selecting for areas with hunters? They might find a carcass or roads or that sort of thing. So what's sort of a general resource selection chapter? So to quickly summarize, coyotes are a novel top predator in South Carolina, and despite our efforts, they seem here to stay. I talked about using GPS collars on coyotes, seeing how their movement changes in response to fawns, and then also seeing how alternative food availability might influence that response. And then I talked about some other projects underway and understand other aspects of coyote ecology. And I forgot to include a slide about acknowledgements, but obviously thanks to my advisor, my lab crew. We have a lot of support from quality deer management, landowners, and DNR, U.S. Forest Service as well. Thanks for your attention.
All right, we can take some questions now. Does anyone have one?
Excellent presentation.
Thank you.
This is Rick Kaminsky.
Hi.
I was wondered about heat maps because, as you illustrated, they show a preponderance of overlap among your study animals. But I would argue that the most suitable habitat, i.e. habitat that enables individuals to survive and perhaps recruit back to reproduce, is habitat where there is not overlap from competitors as well as predators. And we've done this kind of research with with duck nest predators and ducks, and trying to find those habitats that excluded predators. So in your situation, I don't know if you have any fawns that are marked that use habitats or portions of their range outside the range of predators that may cause their depredation. You follow what I'm saying?
Yeah, you're interested in what are quote unquote safe habitats away from predators for the fawns.
Yeah, and I use the term suitable in the context of Fretwell. I don't know if you've done any reading about him. He was the one who defined a suitable habitat, and basically a suitable habitat is one that confers fitness to individuals. So,
sure,
if fawns are the focal prey, but hunters want that prey, wouldn't you be most interested in identifying habitats and then further conserving and managing those habitats where your fawns survive?
That's certainly something we're interested in, I think, and it'd be a great conversation with Mike Muthersbaugh, I think, who's the deer lead on this, and probably looking at where are fawns surviving, where are they living, and that's something I'll be doing. Actually, I didn't mention, but I'll have 2 layers of fawns. One layer will be fawns that died. The other layer will be all the fawns that survived past 3 weeks. And so that's one way, I think.
Yeah, absolutely. I mean, you can do a logistic regression or a discriminant function analysis, and with measured habitat characteristics describing those 2 binary groups. That's all.
Right. Do we have any further questions? All right. Thank you, Alex, for presenting, and Dr. Kaminsky for your questions. We'll move on to our third presenter, which is Jacob Shurba. Jake is a first-year master's student in wildlife biology under Dr. Richard Kaminsky and Dr. Beth Ross. He is studying how the surrounding habitat and internal microbiome of artificial nesting structures affect wood duck recruitment in Georgia and Florida.
Thanks, Crystal. And thanks, Dr. Hagan, for giving me a chance to chat with you a little bit today about my research. Let's see, make sure everything is good here. Okay, so, sorry, make sure my screen is all in order here. So thank you. Yes, I'm working on my master's right now, as Crystal said, currently working on wood ducks in southeastern United States in Florida and Georgia. And so the wood duck is a really special species first off, but in particular, they are a very heavily harvested waterfowl species, especially throughout Eastern North America. And because of this role as a harvestable bird, it contributes to economic revenues very heavily and primarily through waterfowl hunting. And they also, like most other waterfowl, contribute ecological services, ecosystem services, such as seed dispersal, as well as acting as disease carriers and sentinels. Despite this importance, though, they were nearly extirpated by the early 20th century due primarily to market hunting before the proper implementation of annual limits, as well as the loss of very important forested up and wetland habitats that they utilize for nesting purposes. They're defined by multiple authors as secondary cavity nesters. However, they will utilize artificial nesting structures, which we will now refer to as nest boxes. They're also a very heavily phylopatric species, with multiple authors detailing, reporting numbers as high as 79% of wood ducks returning to the same locale for nesting, as well as numbers as high as 42% of wood ducks returning to the very same box that they once nested in for further nesting. It's also thought that these artificial nest structures or nest boxes were very useful in helping recover populations of wood ducks across North America. There's a lot of studies. The one I've detailed is here on the screen. It's from Belrose and Holm in 1994, and they report a project in California utilizing a little over almost 2,300 boxes in California, which they report about a 73% success rate from these nest boxes, which roughly equates to about 9,000 ducklings successfully hatching and jumping from the boxes. And then in a reanalysis of a long-term recruitment study, Hepp et al. In 2019 reports how nest success influences both breeding season survival and apparent annual survival of female wood ducks. Now, wood duck and waterfowl in general, their population dynamics are very, very heavily influenced by recruitment. And recruitment is defined as the process by which young are added to the fall population through reproduction by adults in the spring population. So we've got our nice nest box here, let's say in Florida. We've got a hen with her ducklings who successfully jump and hatch and jump. They grow up to become beautiful like their mama, hatch or migrate out, and then return the following year to this same locale or nest box to rejoin their population. The problem, though, is that these recruitment rates, ones that have been reported, are relatively low. And this is kind of where we're coming into here now for this study. So in 1989, Hepp et al. Reports mean recruitment rates of about 5%. And then Belrose and Holm in 1994 report recruitment rates of about 6%. And like I said, this is low. We want to see numbers considerably higher than this. Usually between 10 to 15% is kind of what we would like to see. Kind of jumping back to this reanalysis of the data from 2019, Hepp et al. Reports an analysis revealing a declining population of wood ducks at about 19% per year. And this indicates that the recruitment of females hatched from nest boxes is insufficient to sustain the nest box population without the immigration of hens from natural cavities in the surrounding areas. Now, this kind of brings us a little deeper into questions about how other aspects of the environment can potentially affect wood duck recruitment. So in particular, no studies have really examined how habitat conditions affect recruitment in particular. The closest that we can see is probably Croft in 2018, who, for his thesis, he investigated how habitat variables and box measurements affect wood duck selection of boxes, wood ducks and black-bellied whistling ducks. And significant results that he concluded was a negative relationship between box selection and internal volume of nest boxes, and indicating that wood ducks preferred smaller boxes, generally with an internal volume of about 15,000 cubic centimeters. And the boxes that were this size generally had about a 91% success, or a 91% probability of being selected by hens. Additionally, he found that the mean percent of canopy cover for selected and used boxes by hens was 41% and 61% for those not used. So wood ducks preferred boxes that were less covered by canopy, which seems a little counterintuitive, especially here in the South where the sun can really heat things up very quickly. Now, multiple wood ducks tend to utilize the same boxes, and because of this kind of increases the chance for detrimental pathogens and bacteria and other creepy crawlies to build up inside of these boxes, which can in turn have an effect on egg viability, because egg viability can decline between this time of laying and incubation inside these boxes. And during this period, microbes in particular can colonize unincubated eggs pretty rapidly. I've listed some pretty general groups. There aren't too many papers that will say exactly what it was that is affecting eggs. They kind of just throw out some general groups of bacteria. And there's authors that say no correlation between bacterial levels and hatching success, but then there are others on the other side that do say that this does have an effect, that microbes do have an effect on these eggs. So this kind of brings us to my work now. And so my primary research question is, how is wood duck nesting biology and ultimately recruitment affected by the nest box characteristics, microhabitat, and microbiome? Now, these are my hypotheses right now. These are, we're not going to focus on all of these just for sake of time, but generally, just to kind of breeze through them though, smaller nest boxes will have a greater use and return rate than larger nest boxes. Nest boxes with less canopy cover will have greater use and return rates. Adult and yearling female recruitment rates to boxes will be similar to those reported by Hap et al. In 1989, and that's about 5%. Nest boxes with increased microbe counts will have an increased number of adult eggs than boxes with lower microbe counts. Nest boxes in Florida will have higher microbe counts and more diverse colonies than Georgia. And finally, nest boxes containing odorous nesting material will have a decreased microbial environment. The nice thing about this project is we do have protocols that have already essentially been established by Croft in 2018, as well as in a pilot study that was run for this project last year in 2019. So for box measurements, we're going to be we take the volume of the nest box, as that was determined to be a significant factor in nest selection. Additionally, we take other measurements such as the direction of the box entrance, the distance of the box entrance to water or ground, and whether or not the box is over land or water. We're also taking habitat measurements at a couple of different times. For a large scale data set, we're taking habitat measurements at the beginning of the season and then again after leaf out. And we chose these times because these are peak prospecting times for hens. Additionally, we're taking measurements at a box anytime it has been initiated. So whether that means that usually means we find one or more eggs in a box. And then additionally, we're taking these measurements at the closest not active box in the same habitat type. So like I said, the first method gives us a large scale habitat data set during these peak prospecting times. This other method gives us a more fine scale data set for habitat surrounding chosen versus unchosen boxes. And measurements that we're taking include canopy cover, visual obstruction, and water depth at the nest initiation and hatch, amongst other variables as well. Now for banding, we hold pretty general banding protocols for the hens. This is essentially a mark recapture study, just a little bit more specialized because we are looking at population dynamics as well. So for hens, we can just throw a band on their leg, take some measurements and let them go. For the babies, we have to be just a little bit more careful about what we're doing because they are too small for a band. So in this case, we use a modified web tagging method in which we place 2 small incisions in the right foot, the webbing of the right foot, and we add this little metal tag. And this is our method of being able to tag these babies, release them, and then come back next year and see how many of these kiddos will return to this natal area. Now for our microbial ecology methods, we are selecting 3 eggs randomly from each box and swabbing an egg with 2 different sterile swabs. We're doing very minimal laboratory work in the field because the majority of this work is going to happen in the fall on campus. So from here, with our swabs, each swab will get plated on 2 different types of agar. We have the triptych soy agar and McConkey agar. And we chose these due to each augers, these growth media, they're the ones that are most recommended by authors and studies that we've used for this, this project, because of these media, the ability of these media to grow a very wide range of bacterial specimens. And these are incubated at 2 different temperatures depending on the type of auger. And then we will check these plates after 24 and 48 hours. We do a 24-hour check just in case we basically don't want to open the incubator and see a lawn of fungus growing across our plates. So that 24-hour check is essentially just to make sure that everything is growing okay. From there, we're going to select isolated colonies from each plate. And this is what we are looking for when I say isolates. We remove those isolates, plate them again, and this will allow us to then perform more close investigations on what's growing inside of these boxes. We are set up to perform DNA analysis as well in the event that that does become necessary. Finally, another kind of smaller aspect of the project is looking at the effects of different types of shavings on the microbial communities inside of these boxes. So a random selection of boxes have been filled with either pine, cedar, or aspen shavings. And this is because there have been some studies that show how cedar shavings in particular have some pretty natural oils that promote antimicrobial growth. So we're going to be looking at the microbial communities from the eggs in boxes that have one of these 3 or each of these 3 types of shavings to see if there's going to be any differences between the microbial growth. We have a lot of collaborators on this study. These are the ones that are pertinent to my location. So Georgia Department of Natural Resources, the Florida Fish and Wildlife Commission, and South Carolina Department of Natural Resources. Clemson University, the Kennedy Center, and the Moores Wildlife Foundation are fantastic partners. It's been great working with them. My technicians and most importantly, my advisors for all the help with the project. And thank you very much. And I am open for questions.
I have a question.
Yes.
So these are cavity nesters, and then now, we've been putting out these nest boxes, which has allowed these guys to rebound almost from the brink of extinction. You're talking about putting pine, aspen, and cedar in the artificial boxes, but I would assume these are not birds that bring any type of artificial anything into their cavities. How is a natural cavity set up for these guys?
Yeah, so the shavings are pretty standard with this sort of box management. So most state or federal agencies that utilize these boxes will fill them with shavings just so that they have something in there. But generally, if they're using natural cavities, it's kind of just whatever's laying around inside the tree. They're not bringing in their own materials, though.
So has anybody looked at nest boxes without any material in there and how the birds respond?
I'm not sure. Unfortunately, I'm not sure.
Okay, thank you.
That is a good question. And Rick may be able to shine some light on that as well.
Hey, I have a question. If you were done with that note, are these only in rural areas or sort of related to that? Are these wood boxes anywhere near urban areas, and does that cause any issues for the wood ducks going into areas where they might be in more danger? The only reason I ask is because last year I had a wood duck in my chimney. I was wondering if there was any reason why that happened.
Wow. Okay. So I'm from suburban Illinois, and I have seen wood duck boxes being utilized in forest preserves in these more urban areas. For my sites, my field sites in particular, I'm in pretty rural Florida and Georgia. However, there is a lot of activity just from hunters, and fishermen being out in the water around these boxes. So I've seen them in both locations. As to why you might have had a wood duck in your chimney, I would say it's an accident. It fell in, but it is a cavity. So it's.
Well, now is the time for me to offer a comment. It was not an accident.
Okay.
Excellent presentation. Excellent presentation, Jacob. But because wood ducks and other cavity nesting ducks like hooded mergansers and common goldeneye and buffleheads are cavity nesters, they'll go down a chimney. They're accustomed to going into cavities that are on the side of a tree, but if a tree, for example, is wind thrown and the top is open, it's called a bucket nest. They'll come in from the top. And when we lived in Canada, my wife told me there was a duck in the fireplace. And I said, really? So I went and inspected it. Sure enough, it was a female common goldeneye who came in through the chimney. And then years later, when we lived in Mississippi, my daughter said, Dad, there's a duck in the fireplace. And I inspected it, and sure enough, there was a female wood duck. So they will go down chimneys. And the other question about adding the nesting period, the nesting materials, that's a great question. We certainly, Jacob, could add a no materials control to your boxes, but I don't think that would address your questions about microbial infection. But as Jacob pointed out, the management regime is to put some sort of shavings in a box to provide a foundation and also increase the height, or decrease the height, I should say, for the ducklings to climb out of the box. So that's why it's done.
Do we have any other questions for Jacob today?
Yeah, I have a question. I was curious, the three types of shavings you were talking about, the aspen, pine, and cedar. Are there any concerns? Are there specific pros and cons among those three shavings that management would be interested in, besides the unknown microbial effects? Are there pros and cons to any of those?
Not from what I can see. Cedar and pine are both ones that are pretty commonly used among managers, and aspen is one that's generally used with laboratory animals, so it comes with really no inherent risk to those themselves. Is that what you're asking, Janelle?
Yeah, that's all.
From what I've read and found, there should be no inherent risk to the birds themselves by using these types of shavings in the boxes. Thank you.
All right, well, on that note, thank you so much for presenting. Of course, I want to thank each one of our presenters for taking the time to present their research to us today. I would also like to thank everyone who joined and provided engaging questions to our presenters today. And with that, I'm going to hand the floor back over to Dr. Hagan.
Thank you, Crystal, for being the moderator.
Thank you.
Thanks, and great job. I'm sorry we had some difficulties in the beginning with Matt's presentation. I'm still not sure what happened, but I'm glad turning my mic on helped, at least for the end. I hope that you will all join us next week as we do the poster sessions for students who are registered for the class graduate seminar course. They'll be sharing their posters in a similar format. I think we have 8 going next week, so it'll be a little shorter per student, and many of them are towards the beginning of their projects. But I think it will be a great time to learn about research going on in the department. All right, I'm going to end the meeting unless anyone has any further questions or comments. Okay, thank you all for joining us.
Thank you.
Thank you.